Hello and welcome to Ethically Speaking, a UCF interdisciplinary series on contemporary moral issues. My name is Steve Kubler and I'm the organizer of this series, and I'm professor of chemistry and optics and founding associate director of the UCF Center for Ethics. In this inaugural lecture, we will learn from Professor Michael Levin about how artificial life is becoming a reality and what the ethical implications are for our world. But before we introduce Dr. Levin, I'd like to take a moment to tell you about this speaker series and how you can participate. I have the pleasure of collaborating with Dr. Jonathan Beaver, who is Associate Professor of Ethics and Digital Culture at UCF and founding director of our Center for Ethics. Together, we developed Ethically Speaking to promote conversations about challenging issues of our time. We encourage you to visit the center's website at ethicscenter.research.ucf.edu to learn more about our activities and to find out about other speakers in this series. We would also like to hear your feedback and your ideas for future talks in Ethically Speaking. Ethics is important to us at UCF, and that's clear from the long list of partners who co-sponsor this series. All of us in this partnership hope you enjoy the discussions in Ethically Speaking, and we hope it cultivates understanding of how ethics provides frameworks for making better decisions, particularly when there are competing values. Today's presentation is being recorded and links will be posted through the Center for Ethics so you can share it with others. At the end of his presentation, Dr. Levin will take questions moderated by Dr. Beaver. You can submit questions using the Q&A box, and you can also upvote questions that interest you. Now, we will turn to Dr. Will Crampton, UCF Associate Professor of Biology, who will introduce our speaker.
Thank you, Stephen, and good afternoon, everybody. It is a great pleasure to introduce today's speaker, Dr. Michael Levin. It's also an honor for the biology department to co-sponsor this important talk series. Dr. Levin is Distinguished Professor and Vannevar Bush Professor of Biology at Tufts University. He is also the director of the Paul Allen Discovery Center and the Tufts Center for Regenerative and Developmental Biology. Dr. Levin obtained dual bachelor's degrees in biology and computer science at Tufts University. He then completed a doctoral degree in genetics at Harvard University, as well as subsequent postdoctoral research in molecular embryology at the same institution. Dr. Levin then held several prestigious academic positions, including an associate professorship at Harvard, before occupying his current position at Tufts in 2008. Dr. Levin's research career has been enormously productive. His publications have spanned the fields of developmental biology, biophysics, and genetics, and have been cited almost 20,000 times with an H factor of 75 and an astonishing I-10 index of 256. His work on the molecular genetic mechanisms that allow embryos to form left-right asymmetric body structures is on the journal Nature's list of 100 milestones of developmental biology of the 20th century. One of the most exciting recent developments from Dr. Levin's research program, which we will learn more about today, is the creation of a kind of artificial life in the form of microscopic, programmable virtual creatures called Xenobots. Dr. Levin, the UCF community is delighted you could be with us today. The floor is yours.
Well, thank you very much for that introduction, and thank you for the opportunity to share these ideas with you all. I am not a professional ethicist or a philosopher, so my goal today is not to provide any answers or to make any specific ethical claims, but what I do want to do is to go over some recent advances in bioengineering and regenerative medicine, which I think have some very important implications for working out ethical principles going forward. And if anyone is interested in contacting me afterwards, or you want to see the details, the deep biology that this is all based on, at these two websites, you can find me. So the first thing I would like to do is to put forward and make explicit the perspective of, in terms of the lens with which I view the world, because I think it's important for us to see these things be made clear. I think that science is the best window we have on the world, and I think that what ought to be should be very tightly coupled to our best possible picture of what is. And so I think that philosophy has to keep up with the science. I also take a kind of engineering approach in my group where we study the natural world, but we believe that a mature understanding must encompass the space of the possible. Not only whatever happens to be true right here and now, but actually all of the adjacent possible facts that could be made to be true as science moves forward. So the main points that I would like to transmit today and hopefully discuss with you all are these. The first is that I would like new technologies to be evaluated from the perspective of opportunity cost of failing to improve life for all. That I will try to point out that evolution offers no support for the common assumption that our world is basically great and all we need to do is not to make it worse. This is a common thing. Many people in ethics discussions of new biology ask, isn't it scary? What happens if this and that bad scenario takes place? And I think this absolutely has to be balanced with the idea that no, we actually have an opportunity and a duty, a moral duty to improve the human condition with research. Okay, so this is a kind of transitioning to an opportunity-based view as opposed to a purely risk-based view. And I'll talk about what it might mean for things to be natural or unnatural and what that could mean for ethics. And in particular, I'd like to describe how bioengineering and chimeric technologies, as well as simply taking evolution seriously, is going to demand a revision to very basic concepts that support theories of ethics. All interesting qualities, intelligence, agency, responsibility, and so on, in fact, lie on a continuum. And we cannot ask which types of systems, these include living agents and new synthetic creatures, are or are not intelligent or are not cognitive in various ways, but we need to ask what kind of cognition they have and how much. Okay, there are no binary categories that are supported by science. And basically, I'll make the claim that we are outgrowing this convenient binary terminology of things like robot, machine, cognitive agent, organism. And these types of binary terms are simply not serving us very well, and we have to move beyond them. So what I'd like to do in this first part is to show you a number of technologies that people have described as scary, and I hope I can show why they are in fact essential. So one thing to realize is that pretty much all biomedical needs, so everything with the exception of infectious disease, things like birth defects, traumatic injury, cancer, and aging, degenerative disease, all of these would be solved if we knew the answer to one question. That question is, how do we control what cellular collectives decide to build? So groups of living cells get together, they cooperate, and they build specific structures, complex organs, whole bodies. If we knew how to tell them to build specific things that we wanted, healthy organs and so on, we could in fact solve all of these biomedical problems. It would be an enormous advance. And the future of our field, I like to illustrate with this, which we call an anatomical compiler. At some point in the future, what you ought to be able to do is sit down in front of this software and draw at the level of anatomy here in the schematic, not from the level of molecular pathways or those kind of details, but actually draw the kind of plant or animal that you want. So in this case, we've drawn this three-headed flatworm. You'll hear much more about flatworms shortly. And if we had a proper understanding of how cells decide what to build, we would be able to have a piece of software that would take this anatomical description, I want some eyes over here, and I want a third head, and so on, and compile that into a set of instructions, a set of stimuli that would be given to cells that would then go ahead and build whatever you just drew. So basically computer-aided design for living forms. You should be able to create any living form that you want, any organ of the body, any novel creature that's never existed before. Okay. Now, I should point out we are nowhere near close to this general capability, although we have a few inroads here and there. But this general capability is still far away. And let's take a look at why it's actually so far at this point. So we all start life as a single cell. So this is a fertilized egg, and this gives rise to one of these remarkably complex anatomies, quite reliably so. But in fact, it's really important to understand how this works, and it's important to know that this is not simply a stem cell biology problem. So, for example, this thing here is a teratoma. It's a tumor that may have hair and skin and teeth and muscle and bone, And what makes this different from one of these wonderful anatomically normal animals is that the three-dimensional structure is missing. The individual stem cells created all the differentiated cells that you would want. It has all the tissues, but it's missing that large-scale three-dimensional organization. So stem cell biology is not in and of itself the answer to this. In fact, this here is a cross-section through the human torso. And you can see the incredible order, the complexity here with all of the tissues and organs in the right place, in the right orientation, relationship with each other, the right size and shape. Where does all this pattern come from? Where is it specified, right? Because everybody starts off as a collection of blastomeres, these early embryonic cells. Now, you might be tempted to say it's in the genome. Well, now we can read genomes, so we know what's in the genome. And in fact, the genome says absolutely nothing about any of this directly. The genome specifies proteins. What's in the genome is a specification of the micro level hardware that every cell gets to deploy. But the genome doesn't show this, any of this information directly. So we still need to understand how does the hardware that's specified by the genome end up making these cellular collectives that know what to make and when to stop growing. And as workers in regenerative medicine, we would like to know if a piece of this is missing, how do we convince those cells to rebuild? And as engineers, we ask one step further and say, what's actually possible to build? Could we get the same exact cells to build something completely different? And if so, how would we do that? So single cells, in fact, are extremely competent. So what you're seeing here is a single cell animal known as a lacrimaria. So this creature has no brain, no nervous system. There's only one cell, so there's no cell-to-cell communication. And yet it handles all of its physiological, its behavioral, its morphological, and its metabolic needs at the level of a single cell, at the scale of a single cell. And remarkably, when cells join together to make multicellular bodies, they didn't lose this intelligence. They, in fact, scaled it up and deployed it towards higher order goals. And I want to show you an example of this. Here is a tadpole of the frog Xenobot laevis. Here are the eyes, here's the brain, here are the nostrils, here's the gut. And these tadpoles need to become frogs. And in order for tadpoles to become frogs, they have to rearrange their face. The jaws have to come forward, the eyes have to move, the nostrils, everything has to move. And it was thought that in some way the genome provided a hardwired set of movements for all these organs. After all tadpoles look the same and all frogs look the same. So if you simply move in the right direction, the right amount, you get a frog. So we made what's called Picasso tadpoles, where everything is in the wrong place. The eyes are off to the side of the head, the jaws are at an angle, the eyes are on top of the head, everything's in the wrong place. And what we discovered, remarkably, was that these animals still make largely perfectly normal frogs, because all of these organs move in novel paths. And sometimes, in fact, they move too far and have to double back to land in the correct orientation. Everything keeps moving around until you get a correct frog, and that's when the system stops. So in fact, what the genetics gives you is a machine that's able to do this amazing error minimization scheme. It's able to meet William James's definition of intelligence, being able to get to exactly the same goal by different means. Despite perturbations, despite starting off in the wrong configuration, it can still get to its goal through novel, unexpected actions. If we had a robotic swarm that was able to do this, we would certainly call it a kind of a collective intelligence, and we'll come back to that momentarily. That plasticity, that ability to get to the correct structure despite perturbations, can be seen, for example, in the structure of the nervous system, where if we make a tadpole that has no primary eyes, but we put an eye on its tail, and all of these are techniques that we use in the lab, so this is not Photoshop, these are real live animals. We put an eye on the tail, and then we use this device to ask if that animal can actually learn to see out of that eye. And in fact, they see perfectly well, so we can train them on visual cues, and these animals can see perfectly well. And you can imagine that this is the basis of sensory augmentation technologies for humans, and we'll talk about that shortly. But this brain, which evolved for millions of years to use visual input from these regions here where the eye should go, now finds itself in a novel configuration where the eye is back here, in fact, connected to the spinal cord, not to the brain. And it has no problem using that data for behavior. So the plasticity is incredible. The ability to handle novelty in living tissue is amazing. And so one of the things that our laboratory is doing is trying to understand how can we control these how can we control these decisions that are made by multicellular collectives? And in controlling those decisions, how can we produce novel anatomies? And this is all part of trying to learn to control anatomy for various purposes. And you can see here a couple of examples. So here are some two-headed flatworms. So this is a flatworm that normally only has one head, but we actually manipulated the electrical conversations that these cells are having with each other, and they built a worm with two heads. There's nothing genetically wrong or different about this animal. It has exactly the normal genome, but the electrical conversations between those cells, we have manipulated them to say make two heads instead of one. Similarly, here's a five-legged frog where we used light to change some of the signaling, some of the electrical signaling properties of cells in the frog's face, and actually we can induce the growth of a whole limb here. So you've seen now a five-legged frog and some two-headed flatworms and a tadpole with an eye on its tail. And really, a lot of people find this scary, right? And about 50% of the phone calls and the emails I get suggest that these bodies are new, they're different, and they're a little bit disturbing. You know, people are used to things that are produced very slowly by evolution over geological time scale. And this kind of thing makes people nervous, you know, that these are very new anatomies. But one of the important things is that we not only have the opportunity to control anatomy, I'm going to claim that we have an absolute moral responsibility to do so. So here are some general statistics, right? 2 million people in the US alone are living with loss of limb, and the mortality in the next 5 years after loss of limb can be 80%. 10 million people die from cancer every year. 50 million Americans are affected with degenerative disease. Every 4.5 minutes, a baby is born with a birth defect, and something like a remarkable 77 billion land animals are slaughtered for food every year. So this idea that our job in bioethics is to simply make sure that we don't screw things up, right? This idea that basically everything's fine and we need to be very careful that we don't make things worse is really, I find, extremely insufficient. And these incredible needs, this global suffering accounts for the other 50% of the calls that I get, which is just an unbelievable range of human patients that require all sorts of solutions that we don't currently have. And so I think we need to focus on the opportunity cost of failing to live up to this moral imperative to use science to improve life for everybody. And a lot of people will always ask, they ask me what keeps me up at night in terms of some of these really wild technologies. And what keeps me up at night is the possibility of failure to live up to our potential, to use the science that we have access to make life better. And so I want to point out that I want to dispel this idea that there's any necessary correlation between what's actually natural, meaning what was the result of the natural evolutionary process on Earth, and what actually is what we want to have happen. This is the basic evolutionary process here. It's very simple. The strategy is this: generate lots of offspring, which resemble but are not perfect copies of the parents. And these imperfections are basically random errors due to changes in the genomes. And then the offspring will compete for finite resources, and only some of them will live to reproduce. And then that goes back up, and they do this again. They generate more offspring, more random copies. Some of them are better suited than others, and on this goes. The important thing about this to note about this process is that what it's guaranteed to optimize is the quantity of biomass that you will observe in any environment. That's all. Nothing in this process optimizes for happiness, for well-being, for any of the things that we as moral creatures should care about. All evolution optimizes for is biomass in any given environment. And I think that this provides a really important lens on this question of what's natural, what's unnatural, and what we ought to be doing, because there's nothing magic about what comes out of the process of evolution. Nothing here is optimized for the things that we find of value. We should definitely be able to do better than randomness. And I want to show you some examples of the kinds of things that may be within our reach. Here's what might be possible. So here's an axolotl. This is a Mexican salamander that regenerates its limbs, its eyes, its jaws, portions of the brain, the heart, tail, including spinal cord, muscle, ovaries, incredibly regenerative. It basically looks like this. If a limb is amputated, or if one of them bites off a limb, which they do to each other all the time, this system will continue to grow, and it will grow exactly what's needed, and then will stop when a correct salamander limb has formed. And so this is one of the most critical areas of research right now, is to figure out, first of all, how to start the process, but even more so, how does it know when to stop? Okay, how does this cellular collective know that a correct salamander limb has formed? But this is the kind of ability that's certainly possible. Evolution happens to have derived an animal like this, a vertebrate animal, a complex vertebrate that does this. And so we know this is possible. In fact, here are those planaria. This is a normal one with a single head. Planaria are the champions of regeneration, because even though it has a true brain, lots of internal organs, all the same neurotransmitters that you and I have, they have this amazing ability. If they are cut into pieces, each piece will regenerate exactly what's missing, no more, no less, and give you a perfect tiny little worm. In fact, that's how they reproduce. They tear themselves in half, and then both halves regenerate. They are so regenerative that they are actually immortal. There is no such thing as an old flatworm. They have no lifespan limit that anybody's ever seen. They appear to have conquered aging entirely. And so then, having seen this and having seen the salamander, one wonders, why is the human lifespan somewhere around 8 decades, 9 decades? There's nothing magic about that number. It could have been 12, or it could have been 300, or we could have been immortal like these other animals. There's nothing magical about the status quo, and it is our responsibility to try to improve it. Now, humans have some regenerative ability. So here, even the ancient Greeks knew that the human liver is highly regenerative. Deer are a large adult mammal that regenerate meters of bone and vasculature and innervation at the rate of about 1.5 centimeters per day. So massive bone growth. And even human children can regenerate their fingertips below a certain age. So all of the limits on human repair capabilities are purely arbitrary. They're the outcome of all kinds of basically satisficing decisions that evolution has made, and the kind of the meandering course of evolution's search through the space of possible outcomes in biology is what we ended up with. But it doesn't have to be that way, and there's absolutely nothing special about where we are with this. Our duty is to control for better outcomes. And I want to show you what that looks like. So this, of course, is the evolutionary process that I started out by telling you, that individual cells can learn to work together towards large-scale goals. Here's a goal: make a limb and then stop, right? Make a normal salamander limb. But that process of scaling up the goal-directed activity of cells can actually break down, and that the scope of the goals that these cellular collectives follow can shrink. So this is a terrible brain cancer called a glioblastoma. And so what you see is these individual cells, they've basically reverted back to a unicellular ancient lifestyle. Every man for himself at this point. They are still very interested in single cell goals, like proliferate, make many copies of yourself, go where life is good, take up nutrients and so on. But they're now completely uninterested in a large scale goal of things like make a healthy liver or a healthy brain or a healthy limb. And it turns out that if you work on this question of how is it that cells cooperate with each other in the first place, you can do things like this. So this is a tadpole. It's been injected with a human oncogene that causes a tumor. But remarkably, even before that tumor becomes apparent, you can already see by using a kind of voltage imaging. This is a way to map out the electrical activity in this tissue so that you can see what all the electrical conversations are that these cells are having. You can already see that here in this region, these cells are going to disconnect from their neighbors. They're going to acquire a really bizarre electrical state and basically uncouple from the network and treat the rest of the animal as just external environment. And once you understand this, you can actually try to gain control of that electrical process. And when you do, so this is our work from a few years ago, when you do this, despite the fact that this oncogene is very strongly expressed, here it is in red, the red staining is the mutated gene that causes tumors, there in fact doesn't need to be a tumor. if you manage the bioelectrical state appropriately. You can reconnect these cells to the network and get them to work towards things like making normal skin, normal muscle, and so on, instead of being a tumor. So normalizing or preventing tumors, not chemotherapy to try to find that balance between not killing the patient and killing the tumor cells, but actually to normalize them. Okay, so that's one kind of advance. Here's another kind. This is a frog leg, and frogs, unlike salamanders, do not regenerate their legs. So having lost a leg, 45 days later, there's basically nothing. But what we've been able to do is to come up with a treatment that triggers a particular, again, bioelectric state at the wound to tell the cells, build a leg here. And when you do that, the leg starts to grow. So just 24 hours of treatment with this particular set of bioelectric drugs kickstarts many months, in fact, over a year of leg growth. And you can see here that the leg is both touch sensitive and motile. And in fact, it becomes a very respectable looking leg, and it grows for a long time. We are now in efforts to transition this to mammals, hopefully someday to humans with limb loss. I have to do an important disclosure here. David Kaplan and myself are the co-founders of a company called Morphoceuticals Inc., which is trying to perfect this technology for biomedical purposes. So that's a disclosure. And there are also ways to address brain defects or other types of birth defects. So this is a normal tadpole. So here's the normal brain. So here's the forebrain, midbrain, and hindbrain. This is an animal that either has a mutation in an important gene like Notch, or it's been exposed to alcohol or nicotine or one of many other kinds of teratogens. And as a result, you can see the forebrain is missing. The midbrain and hindbrain are basically just a bubble. These animals have no behavior. They lay there doing nothing. It's a really terrible birth defect. And what we can do now is, guided by computer models that we've created, we can actually provide very specific drug treatments with drugs that turn on and off the different electrical activities that these cells are using, and convince them to make a brain of the appropriate shape and size. And when you do this here, you find that you rescue the brain shape, the gene expression in the brain, and also the behavior. So they get their IQs back to basically indistinguishable from normal. So we have here the opportunity to use these technologies to and perfect them in these model systems, and then ultimately the goal is to use all of this for biomedicine, both in human and veterinary use, to really relieve the kind of suffering that is out there from all these cases. Okay, so that was part one. Part two of the talk is what I would like to stress is a couple of interesting concepts that are really brought on by advances in this technology. And the first one is this idea that there's a continuum of agency, that interesting properties like intelligence and moral responsibility and memory and all these kinds of things exist on a continuum. They're not binary quantities. And I want to talk a little bit about the plasticity of individuals during their lifetime. Then I want to show you some really novel beings that have never existed on Earth before, and we can talk about what that means for us. The first thing is that when we talk about cognition, we don't only mean sort of a full-blown human-like self-consciousness, right? That's somewhere up here at the end, but all the way down to very simple creatures, you see different kinds of cognitive capacities. So predictive or non-predictive, with feedback or without feedback, purposeful or non-purposeful, and then active and passive, right? So this is a very smooth kind of continuum where there are certain capabilities that emerge, but all along here there are different ways to be a cognitive creature in the world. I want to just show you a couple of examples. This is a slime mold. And what you're going to see, the slime molds are unicellular. This whole thing is just one cell, even though it's huge, it's one cell. And what you're going to see is it's going to solve this maze. Okay, so here's some oats over here, here's some more oats over here. And what it would like to do is to just have the shortest possible path between these oats so that it doesn't take waste energy being elsewhere. And you can see that as soon as it's found the right path, it basically withdraws from all the other parts of the maze that are not part of the solution. Okay, and here it is. You can see here it's pulling away from regions that it knows are not the right part of the maze, And it does this by virtue of, you can see here the flows, these fascinating signals that flow through the structure of the slime mold. And you can see here, it's a perfect solution. Okay, this thing has solved the maze. It found the exit from the entrance to the exit. It's just one cell. There's no brain, there's no nervous system. It's one cell, but it already has the capacity for certain kinds of problem solving. Here it is, and this is our work from a couple months ago. Here it is. It learns about its environment by tugging on the substrate, and it can feel that there are 3 glass discs here instead of 1. It already, in the first few hours of growth, it already has an internal map of its environment, and it will always outgrow towards the larger mass. So it has the ability to probe its environment and then make these simple kinds of decisions. And so what's really critical to understand, what modern biology is telling us, is that if we take evolution seriously, there is no bright line separating any kind of distinction between the cognitive capacities of creatures. So people often ask, a fish or something like this, is it conscious? And then this is or does it have memories? Does it have goals? And this is a bad way of framing the question, because it's not a binary category. There are no binary categories here. We are all part of a continuous ascent along this kind of structure, and all of these things should be asked in terms of what kind and how much. So what kind of cognition, how much cognition, what is the internal world of any given creature like? There's no place to draw a bright line. It's a little bit like the notion of being an adult. There's a convenient fiction that society follows, where on your 18th birthday, bang, you sort of cross over into this line. But we all know nothing actually happens on that day. It's a continuous growth. And so this is actually true for all the things that we're interested in. There are no sharp lines, and we know this even we've known this for a really long time, because if you let's take a concept such as personal responsibility or something like this. If you feel that modern humans have it, all you have to do is walk backwards and ask yourself, at what point, if you think it's a binary kind of quantity, at what point did you have an offspring that had it and parents that didn't? Right? That's what you would be committed to if you thought that there was some bright line. It might be something that develops quickly compared to the geological timescale, but in terms of the biological, in terms of having parents that don't have some sort of cognitive capacity and offsprings that now do, this is biologically it's just not viable. And so we have to understand that all of these things are slowly and gradually changing from more primitive versions to the more complex. And people talk about sometimes the collective intelligence of ant colonies and things like this. It's important to realize we are all collective intelligences. We are all made of cells. There are no one, and in fact, no creature can be a single indivisible cognitive agent. We are all made of parts. And so here's a single cell chasing after a bacterium. You can see, I'll play it again. You can see that here. This is a famous old movie of a single cell. And this is a human immune cell. And our bodies are made of things like this. Our brains, in fact, are a collection of neurons of other cell types, specialized, but still individual cells. And we are, and whatever moral, ethical, personal perspective you have looking outwards, you have memories, you have goals, you have a personality. All of this is emergent from the collective activity of the cells that make you up. And in fact, those cells are the tissues and organs in your body are in fact composed of cells, which themselves are composed of subcellular components that have their own dynamics, and those of molecular components, and so on. So there's this massive multi-scale kind of architecture that biology uses, where there are active processes at every level of organization, at every scale, that have their own local goal-directed activity, and all of this somehow percolates upwards that we still don't understand. And because we are all made of parts, this is why change does not have to be taking place on evolutionary timescales. It can take place during the lifetime of an individual. Here's an example that we've known for a long time. Here is a caterpillar. This caterpillar has a brain. that is perfectly suited to driving this kind of soft-bodied creature. There are no bones, so driving this kind of body is particularly interesting. It crawls and it chews plants. And then this has to transform into a completely different hard-bodied creature, which is going to fly and drink nectar. And during this process, during metamorphosis, most of the brain is basically liquefied. A lot of the neurons are broken down, the synaptic connections are broken, and a brand new brain is rebuilt. So one can ask really interesting philosophical questions. Is it the same creature? You know, if your view of personal identity is as the continuity of body structure, sort of that's the case here, because some of the cells are the same, some of the materials are the same, but many are not. But if your view of personal identity is as contiguous memories, then actually this conserves identity, because it turns out that the butterfly still remembers things that the caterpillar has learned, even though the brain is completely remodeled during this, right? So, and you can learn more about that here. So the brain of an agent can change radically, and one can wonder, what is it like to be an agent? You know, you can ask, what's it like to be a caterpillar or a butterfly, but maybe more interestingly, what's it like to be a caterpillar changing into a butterfly? Okay, and in planaria in particular, they're even more amazing, where you can train planaria to, let's say, recognize a particular set of bumpy patterns to be where they get fed, and then you amputate the whole head, and the tail sits there doing nothing for about a week. Then it grows back a new brain and a new head, and it still remembers the information. Okay, so that information is somehow transferred from the rest of the body onto it's imprinted onto the new brain. And if you think this is just something weird that planaria and caterpillars do, keep in mind that lots of people are developing stem cell therapies for human degenerative disease in the brain. So at some point, probably some point soon, we are going to have human patients with 50, 60 years of memories having chunks of their brain replaced by naive progeny of stem cells. What happens to their personality, to their memories, when no one knows? And so this is not simply you know, a kind of philosophical oddity. It's actually very important in terms of what is going to happen during these therapies. Okay, and I want to talk about now this all of this plasticity is building up to the following: the idea that the body can change, the brain can change, the mind can change, right, to be appropriate to different types of bodies. There's this incredible plasticity of self-repair, of dealing with novelty, behavioral novelty, but also anatomical novelty, right? These are the examples that I've shown you. And one thing that is that's definitely happening is that we are leaving the Garden of Eden in an important sense through a science of chimeric bioengineering. This is a painting of a famous old painting of Adam naming the animals in the Garden of Eden. And at some point, you know, a couple centuries ago, what you could entertain was a worldview in which there was a finite fixed set of animals, right? There were just a certain number of animals, and that's all there were, and they were distinct, and you could name them, and that was it, right? This worldview is crumbling, because as it turns out, we can readily, biology is very accessible to readily mixing contents of different creatures at different scales. You could mix DNA, you could mix cells, you could transplant organs, you could mix whole organisms together and make hybrid swarms. In fact, people kind of knew this already. They knew that you could mix the genetic material of a donkey and a horse and get something different. So this is we already had these kind of hybrids, but now with chimeric bioengineering, the opportunity to make different novel creatures has really rose in an incredible way. And that's because what we can do at any level of organization, so at the level of molecules with nanomaterials and smart materials, at the level of cells, individuals, colonies, we can replace any part of this with either organic, meaning evolved, or cybernetic or designed, or inorganic materials. We can replace any part of this. And this is now a wide range of, in addition to the normal kinds of humans and other animals and so on that are out there, there are now humans with intelligent appliances. These might be neurotransmitter pumps and various other things, humans with brain implants, which control wheelchairs and other devices, and so on. Lots and lots of different types of creatures. And so now I want to show you one example that that we created, and this is collaborative work. It's led in my group by Douglas Blackiston, and my collaborator is Josh Bongard at the University of Vermont, and Sam Kriegman in his group. We asked a simple question. We took some skin cells, liberated them from a frog embryo, put them separately. You can see here in the petri dish. We simply asked the question without any further manipulation, no genomic editing of any kind, just freed from the constraints of the embryo. What would they build, or if anything, right? What would they do? What would these cells do if they had the chance to re-envision their multicellularity? And remember that these are just skin, okay? And so what we see is that overnight they sort of compact together. They don't crawl off. They don't make a flat monolayer. They sort of compact together, and then they start this very interesting activity that I'm going to show you. Once they create this kind of body, they become what we call Xenobots. So Xenobots, it's a portmanteau of two words, Xenopus laevis, which is the name of the frog where these cells come from, and the word biobot. And so here you see what it's doing is it's swimming along, and the way it swims along is it has little hairs, cilia on the outer surface. Normally, these cilia are there to redistribute mucus over the skin of the frog or the tadpole, but they're using it in a different way. They're using it to row against the environment. Okay, so they can swim. You can see they can go in circles. They can go straight back and forth. There's one patrolling. You can see here's a tracking data on a number of them. These are interacting with each other. This one's going in a circle. This one's going kind of on a long trip around the petri dish, and these guys are doing nothing. So they have a wide variety of behaviors. These basal creatures. In fact, one of the ways I sometimes do this talk is I show a lot of videos of what they're doing, and I don't tell people what this is. Okay, and I ask them to guess. And you know, you would never guess that this is a bunch of frog skin that has decided to form a new kind of proto creature. Here's what they do, what they can do in a maze. So this is a water-filled maze. There's no flow, so they're swimming along. So it takes the corner without having to bump into the wall. And over here, some sort of internal dynamics kicks in where it just decides to turn around and go back where it came from. We'll run this again. Here you can see it sort of goes forward. Then it takes the corner, doesn't have to bump into anything. And at this point, just decides to turn around and come back. So they have spontaneous behaviors. Amazingly, they also have regenerative ability. They can regenerate back to their original shape. So here's a Xenobot that was basically cut in half like this. Okay, here it is. And that hinge, you can see here that hinge generates massive amounts of force through 180 degrees to pull the whole thing back into a more proper Xenobot shape. Not only that, if we look at calcium signaling, which is basically a readout for electrical activity, and here are two sitting next to each other, we can see that they're extremely active. This is basically what a lot of brains, you know, if I had a zebrafish brain here, this is what it would look like. It's very brain-like activity. Maybe they're in communication. We don't know that yet. But again, this is just skin. So this incredible plasticity of these cells liberated from their normal environment to give rise to a proto-animal that can reuse its various genome-specified hardware features to in novel ways, in new circumstances. This is a new creature. Now, why do we want to make these? So there's a couple of things. First of all, of course, there are applications for useful synthetic living machines. So if we can learn to control them, maybe they are scraping the plaque off of your arteries, or fixing up arthritic knee joints, or finding tumor cells in your gut or bacteria and so on. So lots of synthetic living machines. More importantly, I think, is that the system provides a kind of sandbox for cracking the morphogenetic code, for asking the question, what is the software? What are the algorithms that allow these cells to join together in a new way, despite when in a novel environment? So really to understand, why is their shape the way it is? Why is their behavior the way it is? And that gets us closer to this idea of an anatomical compiler to solve regenerative medicine, to say, we now understand how cellular collectives make decisions, and we can impact those decisions and grow a new heart or a new leg and so on. The bigger sort of philosophical question is this: where do their goals come from? In other words, they have a primitive goals in the anatomical sense, meaning they make a specific functional structure. They have certain behavioral repertoires. The normal story for where those come from is in any animal is this: well, for millions of years, the ancestors were selected for the ability to do this or that. This is just skin. This skin was selected for the ability to sit quietly on the outside of a tadpole and keep the pathogens out. Okay. And yet, in its own environment, it's able to do these remarkable things. And in fact, if you sequence the genome, all you ever see is frog, right? You don't see anything other than completely standard Xenopus latus. So this question of where do their goals come from? Where do the goals of this novel collective intelligence? And again, it's a collective intelligence because it's the product of lots of cells working together to do this. Is critical because modern science and engineering is currently making tons of new collective intelligences. Here is an example of swarm robotics. These are called kilobots because there's 1,000 of them, and they can be programmed, and they will do certain things that are fairly surprising. You can't really predict them from knowing what the rules of each individual bot are. But this is a kind of existential question for us going forward in society as we make Internet of Things and all these swarm robotics. What is this swarm going to try to do? What are the goals, and where do goals come from? It's a major unanswered question. And the ability to make these kinds of things and to observe the novelty, the novel behaviors and structures that appear are an important way for us to close this knowledge gap. And just to finish up here with a couple of points, everything about biology is, as I pointed out, is swappable. So here's a system where the normal structure of brain, body, environment has been altered. Here is a brain. This happens to be a dish with some rat neurons in it, but the signals of these neurons are processed by computer and fed into a flight simulator game. Okay, and these cells are rewarded or punished based on how well the flight does. If the plane crashes, they get electric shock and so on. And sure enough, what the neural cells learn to do is to fly the plane. Okay, this is now, as far as they're concerned, this is now their body. This is what they're able to do. And so that plasticity, that ability to hybridize living things with virtual bodies, basically with virtual bodies, virtual environments, is really important for the following reason. So here's an example. Here's a gentleman that has no biological arms, but he now has robotic arms that, very much like the neurons you just saw flying the plane, now his neurons are able to drive these mechanical arms. So the plasticity of these are called hybrids, right? Where there's a set of neurons, and they're being used to drive a novel body. Their body is now a vehicle or something else. This is telling us something very important because this forms a continuum. So here you have a human with a little bit of electronics in. So let's say 95% human, 5% electronics, and you would say, right, that's still a human being. There's a couple of things changed, but definitely a human being. Here you have the opposite situation. You have a robot with a few human cells cultured on top. So you would say that's 95% robotics and 5% human cells. That thing's basically a machine. But the amazing thing is that we can fill in now any point along this continuum. If you wanted something that was 50-50 or 70-30, any part of this continuum can now be filled in by a possible living being. So this idea of it is a human, or it is a robot, or it is a machine, this binary sense that we can make a sharp category, simply doesn't exist anymore. Okay, and when one considers the true diversity of possible agents, you can imagine all the synthetics and the cyborgs and all of these different things. It completely erodes these binary kinds of categories that we used to have. In the olden days, you could sort of knock on something, and if you heard a metallic sound, you could say, fine, that's a machine. It came out of a factory. It's going to behave in a predictable, boring way, and I'm morally fine with taking it apart and doing whatever I want with it. Whereas if you do that and it's sort of soft and squishy, you would say that's an evolved creature, and I have to take care of it. That distinction is being eroded. And what that means is that in the future, when we look at creatures in our environment and consider what moral and ethical responsibility we have for them, we will not be able to make those decisions based on what they look like and where they came from. And this, I think, resonates with a lot of other progressive ideas where you simply can't decide what you owe something based on its origin story, whether it got here by evolution or design or some mixture of the two, and what it looks like. We will have to be very, very cognizant of what kinds of minds these types of organisms have. And so Darwin at one point looked out into a riverbank and he said, he was impressed by these endless forms, most beautiful. And I want to point out that that's basically everything he saw in the living world. Everything we see in the biosphere is a tiny little corner of this incredible astronomical option space of creatures that could exist and that are going to exist in our world. We are going to be living side by side with all of these different combinations of biological structures, designed machines, and software. Okay, all of these can be recombined readily. And so our strategy now is that we cannot fall back on this kind of simple criterion that people use in bioethics in terms of, well, how much like a human brain is it, right? That's how we decide what moral protection something has. Where on the tree of life does it fit? Is it an invertebrate? Is it a worm? Is it a dog? What do we owe it? These categories are being blown up by the realization that biology is endlessly plastic and recombinable, and that we need to develop much deeper understanding of what it means to be a cognitive being, a machine. I mean, we are all machines in a particular sense, and so on, and what we owe the various different types of creatures. So I just want to close by summarizing what I've said, that there's new biology that allows us to make all kinds of unusual new creatures. Is fundamentally, it's not about these weird new creatures, it's holding up an interesting mirror to ourselves. It forces us to confront really profound issues which have always been here, but they're obscured by the familiarity of standard animal and plant models. The fact that zebrafish eggs always make zebrafish simply obscures the incredible plasticity of life. And these issues have really deep ethical implications well beyond the simple questions of biosafety. And that familiar categories upon which ethics theories are built are basically crumbling. The distinction between robot, machine, organism, these don't exist the way that they used to, and they need to be replaced. We will, especially the young people in the audience, will be living amongst a huge diversity of creatures with novel bodies and novel kinds of minds. Their origin and physical appearance will not be reliable guides to the moral responsibilities that we owe them, and we need to develop new frameworks that simply don't exist yet. Everything of interest to ethical considerations, including memory, cognition, consciousness, and so on, are on a continuum. We must take evolution seriously, and whatever capacity a human might have that deserves certain kinds of moral considerations and responsibilities, you can trace that very smoothly all the way down to bacteria and beyond, all in increments that do not permit any kind of a sharp line with respect to almost anything interesting. And that evolution optimizes quantity, not quality. And I think I would argue that we have an imperative to improve the status quo. I think surely we can do better than random search. So more details here. If anybody's interested in kind of fuller treatments of all these questions, here are some papers that you might be interested in. I want to thank the people who did a lot of this work. These are the postdocs and the students in our lab. I want to thank my collaborators, people who fund the research that we do, of course, all the animals and the model systems that we work with. And I thank you very much for listening. I'll take questions.
Thank you, Dr. Levin. That was great. We can all give him a virtual round of applause in that awkward Zoomy kind of way. Thank you again for the time. We've got a lot of questions. And I noticed, Dr. Levin, that they kind of come in two forms. So I want to start with the what I'll call the sort of technical and research ethics questions, and then expand out into the bigger picture questions that you direct us to. And folks can use the Q&A to adjust their questions if I'm misrepresenting anybody. But I'll start with a couple of these technical questions. The first question was, how did your team determine what kind of signaling or what amount of signaling was needed to regenerate a frog's limb?
Yeah, so basically what we've been doing for years is trying to understand what kinds of signals drive the creation of body structures in the first place. So what we do is we observe embryonic development, we observe all the signaling that goes on, so we can track the electrical conversations, we can track the chemical signals, And then we build computer models to try to understand what are the cells saying to each other. And in particular, how do those conversations scale up to a larger type of goal? No individual cell knows what a leg or a finger is, but the collective does. And so what we found were some triggers, some bioelectric signals that basically trigger to the cells build a particular structure here. So we can make eyes and limbs and hearts and a couple of other things. And there's many things we don't know how to induce yet, but that's where it comes from. It comes from a study of embryonic development.
Thanks. That's helpful. I think relatedly, we had another question about whether the frog skin cells that create Xenobots continue to lead to the same organisms, proto-organisms, Xenobots, or is there a pretty substantial differentiation every time you run the process? Or the question is, do you end up with differently structured Xenobots each iteration?
Right. It's interesting. It's much like with any other aspect of biology. The large scale is very consistent. So most Xenobots look basically exactly the same at the large scale. But they all have interesting individuality. So their motion types, there's a huge range of different motion types. There's a huge range of different responses to different behaviors. So lots of individuality, but that body type, that Xenobot body type is a consistent thing that these particular cells really like to make, and it's quite consistent.
Interesting. Okay. This next question comes from Professor of Chemistry Eloy Hernandez here at UCF. Dr. Hernandez asks, how can you explain the apparent contradiction between evolution and the self-corrective process you showed in the Picasso's tadpole example?
I'm sorry, I didn't hear the second part of the question after self-corrective.
Self-corrective process that you showed in the Picasso's tadpole example.
Yes. So the idea is this. You can think about the process of evolution, and the most mysterious thing about it, certainly the first time I heard about it as a kid, it seemed wildly implausible, because we all know that if you are building something or you're writing computer code, random changes are much likely to make things worse, not better, right? And so the question is, how in a reasonable time frame do you get all these remarkable capacities that we see in the biological world? And what I think is absolutely required for that to actually work is an important degree of what we call multi-scale competency, meaning that if the eye is off of where it needs to be, it is able within limits to sort of on its own without any genetic instructions to the contrary, it's able to find where it needs to go. That is part of what makes evolution work, because it means if you had animals that were hard coded, and there may be a few that are, but if you had them that were hard coded, anytime that you had a mutation that would change something, everything else would fall to pieces, right? If you had a mutation that moves the eye a little bit, but maybe does something beneficial somewhere else, that animal would be dead. The eyes wouldn't work. The mouth wouldn't be in the right place. The animal would be dead, and evolution could never reap the positive benefits of that mutation anywhere else. The fact that these individual tissues and organs are self-correcting in anatomical space and physiological space, in transcriptional space, they have a lot of capacity to make up for differences in their environment. I mean, some amazing examples I could give you where that means that it acts as a buffer. It means that evolution can now explore all kinds of potential changes, because things will try to get their job done even when their environment is different. So if the eye ends up on the tail, no big deal. It's the optic nerve still knows how to find the closest large nerve fiber and synapse onto it. And so the brain knows, well, if information comes from the spinal cord, I can still use that. It doesn't have to come through the optic nerve. So that plasticity, I think, is actually a crucial part of evolvability. It's what makes the process so effective and rapid.
Okay. So relatedly, John Whitesample, a professor of biology here, asked the question. He says, fascinating research on understanding fundamental biology, which may have myriad applications. Any thoughts on the mass extinction of species that have evolved over millennia versus the creation of novel organisms?
Yeah, boy, that's a great question.
Now we're going bigger question.
Yeah, I don't really have too much to say about extinction, but I will say that one of the biggest issues with the open questions of evolution is the so-called arrival of the fittest, right? I think we understand selection fairly well, and you can sort of see how how the environment and competition would winnow out things that are suboptimal. But where do body plans come from in the first place is a really interesting question. And one of the things that I think the work, for example, in the Xenobots is telling us is that there are aspects of this that we simply do not understand, because unlike every other creature on Earth that I'm aware of, these things do not have a lengthy evolutionary backstory of selection towards their shape. This thing basically put itself together in 48 hours in front of our eyes under completely novel circumstances that in this lineage did not exist. And so the ability of this novelty, the ability of if something happens to the genome and now the mechanisms telling the system where the signaling molecule for where the eyes are supposed to go is now off in the wrong place or whatever. All of these are viable organisms. That's the amazing part about this, right? The frogs with the eyes on their tails, the six-legged frogs, the two-headed worms, all of these are completely viable. They may not be ecologically competitive with others, but that's okay. They're all viable creatures. So there's this incredible source of variety and variability of living things. And I think that's really important for the appearance of new species. In fact, something I didn't have time to mention, but those two-headed worms are in fact a kind of new species, because if you cut them into pieces again, they continue to make two-headed animals. So even though their genomes are completely normal, no changes to the genetics, the question of how many heads you have is actually stored in an electrical circuit that we've modified to make two-headed animals. That circuit has its own memory. They will continue to make two-headed worms in perpetuity, as far as we can tell, forever. So, is that the creation of a new species? I don't know. Much like everything else I've shown you, it kind of erodes, these classical definitions. But it's a permanent new line of animal.
Yeah, my brain is spinning now about connections to sort of the de-extinction debate and whether bringing back an extinct animal is the same thing as creating a new species. And yeah, super interesting. We've got maybe two more questions about the big picture biology things, and then I'll ask you a couple of questions, big picture about ethics specifically.
Yep.
So the first of the biology related questions comes from Mason Cash, who's a professor of philosophy here. Dr. Cash says, you said that there are no bright lines between different levels of cognitive sophistication, but many argue there are some really bright lines, especially in cognition, responsibility, self-awareness caused by environmental factors like language. A human child raised in a social world without human languages, like raised by wolves, lacking a history of development in that cognitively enhanced environment, ends up with a dramatically different cognitive capacity.
Sure.
So development of cognitive capacities relies on a very strong degree of environmental factors like language that scaffold cognitive development. Languages are a ratchet that help lock in learning and changes.
Yep.
So the question is, are you understanding the ways biological and evolutionary sculpted development has been fine-tuned by developing and or evolving in a context that includes the requisite environmental scaffold?
Yeah.
So will it be possible to replicate that kind of development?
Yeah, no, it's a good point. I don't disagree with anything you said, in the sense that what I certainly don't mean is that there aren't great transitions. So there are qualitative leaps in cognitive capacity, and I showed you kind of a ladder of one possible ladder, and if I totally agree that language is such, and so on. What I mean by no bright lines is that people often, when they're not careful, they often talk about these things as if they were binary categories. And the reason why that doesn't work is because what that asks you to, I mean, certainly creatures with symbolic language are different than those without, but what you are not going to find is a single point in evolution where here were some parents that did not have feature X, and then bang, their offspring, right? Here's the change, and then their offspring now have full-blown language, right? We don't find that. So even though certain cognitive capacities might have evolved rapidly is again only rapid on the geological time scale. If you actually look at the biology, many generations it takes, and so there's nowhere where you can put a binary that that creature has it, that doesn't have it, and then the offspring do. And so that's all that I'm trying to put an emphasis on, is that all of these things are the biology is changing quite slowly, right? And the result is often, again, it only looks rapid on a geological timescale. Actually, all of the changes are quite piecemeal, and that's why I think we have to be really careful because whatever kinds of let's say responsibilities or in fact privileges that we give to a particular kind of creature, we have to ask, what do we think about very, very similar creatures which we can make or which already exist that are all in the space of that adjacent possible, right? We don't, you know, and that's really the thing, is that all of these things are not like crisp categories. They're all kind of a cloud of other creatures that are similar to them in some ways and different in others, whether they've been modified or just evolutionarily different.
Yeah, it seems like the sorts of questions and responses you're working with in terms of artificial organisms or artificial life mirror the same sort of work that's going on across lots of other species, right? There's all kinds of really interesting work on cephalopods and intelligence and cognition. Same with like tree communication through chemical responses to predation and all sorts of things, sort of seeing cognition, or whatever you'd like to call it, across this gray sort of scalar model. And sort of it seems like you're coming to those same questions through the work on Xenobots that other folks are coming to working on existing organisms.
I think that's true. I mean, so evolution is just a slower version of what's now being done in synthetic bioengineering. Evolution has been telling us forever that these creatures are part of a slowly changing continuous lineage, right? And we are just expanding that into very, very much more rapidly expanding it into multiple directions across the space of all possible creatures.
Interesting. Okay. The last of the biology questions comes from Professor Will Crampton in biology. Professor Crampton asks, the physicist Richard Feynman once said, "We can't make what we don't understand. One thing humans have never managed to do is make something living out of something dead. Your wonderful artificial Xenobots are made from the cells of a frog, while Craig Venter's synthetic Mycoplasma laboratorium was made by injecting a man-made genome into a bacterium stripped of its own genome. In your opinion, is it conceivable that humans, at least in our lifetimes, could create a truly synthetic, self-replicating organism from scratch, one without parts made from other living organisms?"
Yeah, well, the simple answer I would say is yes. I think that's absolutely possible. The deeper issue here is that, much like with many other terms that have been eroded by modern progress, I'm not exactly sure what living means anymore, and I'm not alone. There are lots of people that work in exobiology, right, and try to understand. Okay, if we were to examine life on other, if we were to examine other worlds, what is it that we're actually looking for to say, that's life, you know? And people, there's lots of really interesting work on what's called metamaterials and smart materials, and that. Given that all, you know, all of life is also a continuum, the bottom end of that continuum from bacteria and sort of lower down all the way melds very, very continuously into material science. And so figuring out where you're going to put a line that says that, and this, I've heard this before, and it kind of drives me crazy. People say, now that's just physics. Well, guess what? Everything is physics, but there are different kinds of physics that are exploited in different ways. And I think there's going to be a very smooth gradation between things that we clearly have always recognized as living and things that look like just physics. There are some amazing examples of, you know, oil droplets that also solve mazes and things like this. And I think I absolutely think all of this is going to, those two fields are going to merge, I think.
Interesting. I know we're short on time, so I want to ask you just two last questions to wrap up. One is very big picture. This comes from Rabbi Sanford or Shansky, who writes in the chat, "You made no mention of religion, from which most American and Western ethics is derived. What should be the role of religion in determining the ethics of the modification of typical life forms and the creation of new artificial life forms?"
Yeah, wow, that's not a small question. Okay, I'll take a stab at it. So this is my belief, that whatever the role of religion is going to be, it absolutely has to be tied closely to what we understand about the natural world. Okay, I think to the extent that religion is giving us useful frameworks for action, it has to be consistent with what we know, and that's fine. There are, as far as I can see, there are many compatible ways to understand religious precepts in ways that are compatible with the science. But I think all of it has to work together towards a kind of life positive outcome, right? I think however we interpret religious thought, I think it has to be in a way that is going to be for the benefit of society, and that means, to my way of thinking, that means it has to be firmly grounded in the reality of what we know. So if there were religions that were to say that there are only, and this is actually very interesting. So I, in fact, have an appointment next week to talk to the rabbi at Tufts about very similar issues, and I've worked with some Buddhists and so on. About the status of these various creatures, because actually if someone did have a religion that said there are only X number of creatures and that's all that there are, that clearly now seems to be incorrect, right? It's just simply incorrect. And so if we're going to have useful religious framework to guide our activity, it has to be brought up to date with what we now see as possible. We have to take these creatures seriously. We have to honor their ability to suffer, and we have to sort of modernize as much as necessary those ideas.
Thank you. I know that was a big question. The last one I want to ask is to circle back around really briefly to, in a way that I wish we had more time on, to your central thesis, what I took to be your central thesis, which is that we've actually got a moral duty to improve the world through this type of research.
I think so.
As opposed to taking a risk-based view of technological development and scientific progress. But my mind, when you stated the thesis, my mind immediately went to all of the litany of ethical missteps that we've taken in scientific development, both on the biology end, but also on information systems and destructive systems, and you could go on and on. So I wondered if you could just talk with us just very briefly again about that central thesis and how you see that, I don't know, risk-benefit analysis playing out.
Yeah. Look, I think it's for sure true that you have to do the risk-benefit analysis as much as possible, because it's often hard, because we often don't see all the implications of our work for any specific case. So I think it's essential to think about the possible misuses on all of this stuff. But if we take the 30,000-foot view and ask broadly, how often is it the case that we would have been better off without scientific inquiry? Okay, and I suppose this could be debated, but I'll just give you my personal view. It seems to me completely convincing that overall science is a good thing. And much like anything else, we screw it up from time to time and misuse it in various ways. But overall, I can't think of a pre-scientific time period, in fact, in any time period prior to now, that I would like to live in versus now, in terms of, and I know other people more competent than me will have statistics about, what it was like and how it was to live in these other areas in terms of violence and just the ability to me, it's the ability to be more than the biology. Right. And I think this gets back to the previous question. Survival is fine, but the ability to actually live in the world where you have free time and the capacity, the freedom and the capacity to sort of explore and exploit your creative gifts, so that you're not spending all your day digging for roots. This is how many tens of thousands of years of that do we really need? I think that science lifts us up out of mere biology to more sort of more exalted ways of being, and I think that's essential. And I think we are going to mess it up from time to time, and we have to do our best. But I don't see any way, especially with the medical suffering in particular. People often will say to me, "Oh, that's too much," and I'll say, "Well, pick a point, because at some point, putting a splint on a broken bone was maybe considered too much, right? Was that too much? Blood transfusions, I suppose some people think that's gone too far. Now we have brain surgery, we have all these things. I don't see any moral way to tell any of the people that call me to say that my kid has this problem, I've got a spinal cord injury, I've lost a limb. I don't see any moral way to tell any of these people that, we've decided that enough's enough. We're just going to sort of like stop here. I think that's a complete non-starter in an ethical sense.
Yeah, thank you very much. There's endless questions we could talk about, from concerns about justice and access to the sorts of technologies you're talking about, But I'm really grateful, Dr. Levin, for your time, and to everybody for asking really excellent questions. Thank you so much. I'm going to turn things back over to Dr. Kubler to wrap us up. Thank you again.
Thank you very much.
Thank you, Dr. Levin, for an intriguing and thoughtful and provoking presentation. And thank you, participants, for your outstanding questions. A link to this presentation will be made available at the UCF Center for Ethics website, so you can view the recording again and share it with others. And there you can also find information on upcoming talks in Ethically Speaking. Our next presentation will be on November 4th rather. Dr. Brian Quick, Assistant Professor of Philosophy and University Studies at Portland State University, will give a talk entitled Biotechnology and Global Health, Lessons from the COVID-19 Pandemic. Do sign up for that webinar by visiting our webpage at ethicscenter.research.ucf.edu. You'll also find information about the center's other activities, as well as contact information to provide feedback and ideas for future talks. This series has come together with the help of many people, and I'd like to thank the Office of Instructional Resources for technical support, Nicole Wills, Heather Gibson, and Matt Dunn of the College of Arts and Humanities for promoting our event, and Dr. Will Crampton and Dr. Jonathan Beaver for helping with introductions and Q&A. This series has resulted through generous support from many UCF colleges, departments, and units listed here. And I'd like to thank these partners and many individuals in those units, too numerous to list, who have helped bring this together. Lastly, thank you for your participation, and we hope you will join us for future presentations in Ethically Speaking.